Hello everyone. I am Harris. I'm from Team 6 and we call ourselves as Spartans. And the reason for this because in the past 3 days we've been working on this thing called Sparta or Sparkdiff annotator. And I'm the lead of this project and mostly focused on the front end part of the project. We have Amo, he's working on the database and also back end. We have Akram, he's working on the database and documentation. We have Archie, and he's working with the back end and also documentation. So for those of you who are familiar with Spark Portal, you may have realized that to this date, we only have a way to preview a JPEG 2 file using Biolucida Viewer. But what about TIFF? TIFF is a very common microscopy image file format. Unfortunately, right now for TIFF, we don't have a way to actually view it in the Spark portal. So we have to download it, wait until our download finishes, and then we can open it in our local computer. So this is where our solution comes into play. It's called Sparkta. It's a TIFF viewer and also annotator for Spark. So the TIFF viewer was built on top of OpenSea Dragon and dip some images. And the reason for this is because we want to preserve the resolution without having the user browser broken because of the file is being too large. And using our viewer, users can view the TIFF images, zoom in, zoom out, and use full screen if they want to investigate further of the image that they see. We also provide the custom annotation, and we built this using Annotatorius. And using our custom annotation, user can create, save, and share the custom annotation directly on the images. So this is how it will look like. And now every time you see a TIFF file, you will also have an extra action button. And if you hover over it, and if you click that button, you will be directed into our Sparta viewer. This is the overview of the Sparta viewer. On the left part, you have the toolbars of the things that you can do with the viewer, and you have the image on the right part of this Sparta. Sparta offers different functionalities. So for example, zooming and panning. And if you want to explore it a bit further, you can also go into full screen mode. You know that now you have a very large screen, so you can identify detailed cells in the tissue image that you have. Not only we allow the user to preview or to view the file, but we also allow user to create annotation. Maybe when they see the microscopy image, they find it interesting and they want to keep track of everything that they see in that image. And using Sparta, you can create annotation based on that. You can add command, you can add tags, and the annotation shape is not only rectangle, but it also comes in different shapes. Like for example, you can use ellipse, you can use polygon, and for some specific tissue that you want to highlight a bit detailed, you can also use our freeform annotator. We also care about reproducibility and reusability, so this is why our Sparta has download and upload functionality. Download, you can download the annotation that you created, and by upload, you can upload it later back when you want to see the annotation that you created again in the same image. So the goal is to eventually integrate the Sparta to Spark Portal, just because we have a very limited of time. What we think is we created a simulation on how everything will fit into the Spark Portal environment. So we have the Sparta front end. This is basically just some few components try to mimic the current Spark UI, and also we have the Sparta component inside of it. And also we created the backend using Flash app. And the idea of this one is it will be the one that serves the information that is being needed by the Sparta. We also created a mock database, and inside of it, we have the annotations that the user created to actually mimic how the user can create different type of annotations. We believe that Sparta will be impactful, firstly, in a way that it will improve the user interactions. So if previously the user has to download large image and wait for it, now with Sparta, they can directly preview the image. Or if previously the user has to download some custom software and has to pay for it to annotate images, now they can just use Sparta in the Spark portal. We also believe that this will foster collaborative community, especially because we have this upload and download annotation functionality. So user can share the annotation to their friends if they want to share some insights and highlight important features directly on the Spark portal. Sparta also supports the FAIR principle. So we believe that Spark enhances the fairness of TIFF images on Spark by making them more findable in a way that TIFF files now can be better explored, accessible in a way that TIFF files on Spark can be arranged, can be accessed anytime, anywhere with no need to download, interpretable, because right now it's the annotation will be understandable and shareable to other people, reusable because we allow the download and upload of custom annotation so users can share this one to their friends and they can see what kind of thing have been annotated so far. We have a lot of ideas, to be honest, that we want to work with Sparta, but because we have a very limited of time, we try to prioritize on what kind of things that we need to get done in like 3 days. So we really want to integrate Sparta into Spark Portal. We want to support multi-dimensional, multi-channel, multi-slice TIFF files. We also want to change the tile generation from static to dynamic now for deep zoom files. And for annotation, we want to add more features. Like for example, maybe we will allow the users to create their annotation to be public so more people can see it, and it will definitely offer more collaboration in the future. That's basically the Spark that we created, and we believe it won't be possible without the help from the mentors, the open source community, especially OpenSea Dragon, because the Spark that we created relies heavily on that library, and also the flat icon creators because we've been using flat icon a lot. Yeah, thank you everyone.
Welcome to the sparc.sci presentation. The SPARC project unites researchers from 60 groups representing 90 institutes in their exploration of the autonomic nervous system towards the development of therapeutic neuromodulation. Visualizations are a core part of this process, allowing scientists to interpret data and draw inferences. It is crucial in enabling collaborations and the communication of results. The problem facing Spark users at the moment is that there is no tool available for researchers to easily generate and disseminate visualizations across platforms. Powerful tools do exist, but they are not accessible on all platforms. This limits the interoperability and reusability of existing tools, making it difficult to share visualizations across different platforms and software environments. The solution we have developed is Spark Scaffolding in Python, or sparc.sci, a tool that enables reproducible and distributable visualizations alongside insightful analyses, all in accordance with FAIR principle. Central to this project is a tool that allows the user to easily manipulate, visualize, and export scaffolds using a Python interface in a standard format compatible with visualization and computational modeling tools such as ParaView, OpenFOAM, and Phoenix. This can be run within OSpark with access to data through Pensive and provides interactive visualizations that support generation of scaffold level or landmark level metadata to enhance the discovery of visualizations and increase the value of existing data. Included within sparc.sci is a series of tutorials to showcase its capabilities and to ensure that it is accessible, lowering the technical barrier to generating visualizations. The tutorials start with generating a visualization from searchable public datasets available through Pensive. We then use sparc.sci to derive insights and label the constructed scaffold classes with searchable metadata tags. Finally, we show how sparc.sci can be used alongside existing tools and import new experimental data into the scaffold class. sparc.sci supports the Spark research community by making visualizations easy. It reduces the technical barrier for researchers to visualize scaffolds across multiple platforms and draw meaningful insights from data. It enhances the value of existing datasets and supports the generation of insightful visualizations within the Spark community. On behalf of the sparc.sci team, thank you.
Dear listeners, dear judges, thank you for watching this presentation of our submission for the Spark Codeathon 2024. Our submission is OSpark Hub, an extensible ETL framework for streamlining the Spark ecosystem. The Spark databases provide multiple datasets, such as anatomical models, neural connectivity data via the SCAN database, physiological and electrophysiological data, genomics data, and many more. To supplement this, OSpark is a platform provided by Spark, which consists of services, code templates, and modules to manage multiple Spark data types. However, there are not tools for all data types. As a solution, we decided to create the OSpark Hub framework. It is a collection of Spark pipelines, modules, and applications developed to fill the gaps in the OSpark platform. It integrates a pipeline to navigate the datasets available on the Spark portal directly from the platform. Depending on the data type, free applications are available to access and use the data. We can use data from the SCAN database, anatomical models from the Spark portal, and genomics datasets from the portal. Additionally, multiple modules were developed to process various data types. The developed OSpark apps are deployed on the OSpark platform and submitted to be publicly available. I will now share some video demonstrations on the app's usages. The first application requires a VTK file as input. Three options are available. The user can either import their own VTK file by clicking the upload button. They also have the option to visualize the Spark portal datasets with the ability to choose a target species and organ. After clicking on the submit button, a list of corresponding Spark datasets will be displayed. After defining the preferred dataset ID, the 3D model will be generated. Additionally, a demo option is available to showcase the app. As you can see within the next few seconds, we can observe this model from any angle, as holding left click rotates it, the mouse wheel zooms in and out, and right click moves the model itself. The next app requires transcriptomics data in the form of a CSV file as input, which can be uploaded using the upload button. Once again, a demo option is also available. The outputs include an expression profile analysis and visualization in a volcano plot. Additionally, an oncology analysis will be performed and the results will be illustrated as a bar plot. This last app shows access to the SCAN database. The user should define the target species and organs to investigate the neural connectivity between the chosen regions. The results include a table, a simplified body map illustrating the neurons between the regions, and a simplified plot illustration of the intermediate regions. The OSpark ecosystem provides a limited number of reusable services. With OSpark Hub, we felt the best way to contribute towards this framework is by modularizing our components and expanding the library of services. Accordingly, we have now implemented 6 new services covering a wide variety of common functionalities required in constructing your own studies. Similar to how we illustrated in the video earlier, here we see how these services can be plugged into one another to allow us to fetch a scaffold file from the Spark portal, convert it into the required format, and then visualize it. Another example, the users can upload their own data, convert it if it is not in the correct format yet, and inspect it as well. And in this final example, we once again see how the Transcriptomics Explorer service allows you to readily view and explore gene data. To wrap up what we have seen in this presentation, OSpark Hub is an extensible ETL framework for the Spark ecosystem that offers users the ability to access a large variety of datasets in different formats and gives them the ability to find, analyze, and visualize the data they need with ease through the apps and modules that OSpark Hub offers. OSpark Hub has been conceptualized and developed by us, Shailesh, Heba, and Finn. And with some final acknowledgement, we would like to thank you for listening.
Hi, my name is Max. I'm going to present on behalf of Team 8. We developed sparc.RL, which is a reinforcement learning pipeline which should make medical device control easy. What motivated our work? Closed-loop control of medical devices can improve therapeutic efficacy of, for example, neuromodulation. Control algorithms are typically developed based on in silico dynamical system models obtained from experimental and clinical data as available on the Spark platform. This requires robust controllers which guarantee safety performance, and therefore reinforcement learning algorithms are gaining rising attention. However, unfortunately, reinforcement learning requires expert knowledge and has a very steep learning curve, and therefore it's still underused in a biomedical field. sparc.RL should solve those issues. It has the goal to combine the best number of biomedical data available on OSpark and the computational platform of OSpark in an easy-to-use tool for engineers without background reinforcement learning. For the Codeathon, we chose one specific use case, which would be vagus nerve stimulation for cardiovascular applications. We developed a closed-loop vagus nerve stimulation controller using the sparc.RL pipeline and OSpark that allows to treat elevated resting heart rates. We developed a local standalone tool to load and pre-process a time series Spark dataset on cardiac effects of vagus nerve stimulation in behaving pigs and train a surrogate model on that. We also used a synthetic dataset generated by a model of the acute cardiac effects of vagus nerve stimulation available on OSpark and trained another surrogate model. We then chose the second surrogate model and employed the sparc.RL pipeline on OSpark to train a reinforcement learning controller to lower the heart rate to different levels that can be specified by the user. We then evaluated the control on the surrogate model. What you can see here is a standalone client that we developed. You can pick here available time series data from the Spark platform. We can view the dataset description here, and also we produce a suggestion on how to use the dataset in reinforcement learning using a large language model. Once you pick the dataset, you can download it to your hard disk. Next step, you can select the file from the dataset and pre-process it. You can visualize the pre-processed data, and if you are happy with the pre-processed data, you can move to the next step, which would be to design a deep neural network architecture to train your surrogate model. You can also specify your training parameters, such as the optimizer, the learning rate, or the selected batch size. And you can visualize the model architecture you generated. And once you're happy with it, you can run your training. Let's move on to the sparc.RL pipeline in OSpark. This is a fully integrated reinforcement learning pipeline. You can upload input data to train your surrogate model using the sparc.RL train surrogate model node. This can be either data generated through the standalone client that we have shown you before, or really any data generated by computational models. Once you have trained your surrogate model, you can move on to reinforcement learning training. You can upload a configuration file to parameterize your reinforcement learning training using the sparc.RL train agent node. And once you have trained your reinforcement learning agent, you can evaluate it on the surrogate model Let's move on to some results. What you can see here is the heart rate response to vagus nerve stimulation on a certain stimulation intensity. The blue line here is the ground truth data. The orange line is the prediction of the surrogate model, and you can see that the surrogate model captures the system dynamics very well. Here you can see the reinforcement learning agent training in terms of loss over training iterations. So we had 30,000 training iterations. You see a very nice convergence of all the losses over the iterations, suggesting that we found a really well-trained controller. We then evaluated this trained controller in a heart rate tracking task over 1,000 seconds, in which we varied the set point heart rate every 50 seconds, and the controller was able to really track very well the different set points. Would like to give a big shout out to the Spark Codeathon mentors, especially the ITIS team. If you're interested in the project or have any questions, just shoot either me or John a message by mail.